we get experimental science. We're curious, non-judgmental.
I'm an antimicrobial peptide, sometimes NMD. That's not to say that I couldn't be, but NMD, I will say that you have lots of antimicrobial peptides, as does Smikes, as does Pericles, as does many people in the chat. No, not you are one, you have them. So, antimicrobial peptides, they are called AMPs. That is the antimicrobial peptide. So, the words, I think, are, if we break it down, it's pretty straightforward. A group of peptides makes a protein. Anti means against. Against microbes, you have peptides. It is that straightforward of a nomenclature. It's one of those things that really is hard to dissect because people just use that acronym of AMP all the time. And so if you don't know what the AMP stands for, you'll have no idea what the actual molecule is supposed to be. So this molecule and series of molecules prevents infection by bacteria as well as fungi. And they do it a couple of different ways. There's 4 ways that they can wreck a cell via messing up their cell membrane. And there's 3 ways that they can mess a cell without rupturing the membrane. So there's a total of 7 different ways that this molecule can act. So what antimicrobial peptides will do, so just a couple of quick hitting facts on them. They are anywhere from about 10 to 30 amino acids in length. So they're small. They're on the small side. What's also really cool about them is that they are double. They are called amphiphatic. Oh my god, I don't know how to say the word. So what that means is structurally, this is a very simplistic model that I'm drawing out. This is our AMP. Part of it is water loving. That means hydrophilic. So it works totally fine in water. And the other part is hydrophobic, or water fearing is essentially what the definition of it would be. And so why is this cool? It is a mechanism by which these molecules can wreck an invading cell type. So here is our AMP, one side, and here is the other side. So Gwen, it's a cell membrane, what I'm trying to do. The reason drawing this out is because these AMPs, there's 4 ways that it can wreck a cell by messing up its membrane. The first model is called a barrel-stave model. And essentially what ends up happening, these molecules insert into the membrane. And as they insert, they actually cause a break point. They break the inner and outer membrane. And what you end up having is water can rush into the cell and cause the cell to pop. So that is the barrel-stave model. The second model is called toroidal model. This model does a little bit something differently. You still have the antimicrobial peptides are in here, but they do something a little different in terms of the actual membrane. So if this is the membrane here, what they actually do is they cause a curvature to form. So they actually reform the membrane like this. So the membrane isn't popped, it's just separated out. It's not like a full-on hole, it's that they're still reconnecting the pieces. It's now they're making a much more of a controlled artificial opening. And again, the same thing can happen here, is that you have water will rush in and again pop cell. Your next one, number 3, is the aggregate model. And what the aggregate model does, it ends up just wrecking the plasma membrane. So remember, if we actually zoom in here, what this membrane looks like is that there's these phospholipid little heads and there's these tails pointed inward. And then there's the other side is doing the exact same thing. And these tails are running parallel to one another. What this aggregate model does is you get these AMPs and they just insert all over the place. And as they do that, they break the connections that the phospholipid bilayer has. And so it's no longer going to be nice parallel lines, but instead what you'll end up having is something like this. And again, with the amps are in the mix in between here. And so because the plasma bilayer is messed up, you get, again, water flowing in and yet again, pop cell. The fourth model is called the carpet model. And it's, again, a way of totally wrecking the plasma membrane. But here, the big difference is that you have these antimicrobial peptides end up coating the plasma membrane. And so then you end up cut into the membrane here and here. There's the opening that we're building. You'll actually then have more of these amps coating every region of potential opening. And so it prevents the cell from fixing itself. And so it's each part of it is totally covered. And then the water comes in. Those are the 4 ways that it can wreck the membrane. Then there's also 3 different ways that it can end up wrecking internally. what you can end up having is that this antimicrobial peptide goes through the membrane and goes into the cell. And normally, we have the simple process of you going from RNA to DNA, protein. That's your standard synthesis metric. Normally, what can happen in this process is that you'll have enzymes, and these enzymes will act on certain proteins. But what the antimicrobial peptides can do is they block this step, so they block folding, so the proteins don't fold in a correct manner. They can also inhibit the enzyme, so stop the enzyme, which has essentially the same function as inhibiting the folding event. And then they can also come in here and increase the level of what is called lyse, and that causes it from internal cell lysis. So they will induce the small molecules that causes these cells to pop. Again, not using the physics of water. Lysis just means the pop. So you can have a cell undergo cell lysis if it has like that change in water pressure, which is what would happen if you're using the membrane approach, because that's just water rushing in because the cell is no longer in equilibrium, and so everything just rushes in and causes the cells to pop. So these are the 7 different ways these antimicrobial small molecule peptides can act to drive something like this to happen. Peptides, they are part of the immune system. Yes, Mikes. They can also be acquired and used as a treatment method. So there are antimicrobial treatments that were derived from animals as well that you might be able to take, again, for antifungals or antibacterials. And so why these are cool papers is because they describe new antimicrobial peptides or new way of finding them. And so from a health point of view, if you identify these antimicrobial peptides, you can actually use them for potential treatment options. Does that make sense, y'all, as to what the heck an antimicrobial peptide is? That's the foundational piece. There's multiple ways that they can mess up invading cell. Again, it can be fungal cell or bacterial cell. As far as we're aware, they're not directed against viruses. So just again, the fungi and the bacteria. There's 4 different models by which they mess with the membrane, and there's 3 ways by which they mess with the inside of the cell without directly acting with the membrane.